Thank you, Rudy, for being our speaker today. I've actually known Rudy for probably close to a decade now. So Rudy actually got his start here at UCSD. That's not where we met, but as an undergraduate researcher in labs of Victor Mazet, Nicholas Webster, and Brian Pelnick, getting a really early start on doing research on bacteria, which are awesome, even though I don't work on them anymore, still very old love of mine. And then he continued on to the University of Washington, where actually I met Rudy, working in the lab of Ferric Fong, tackling some really interesting projects on nitric oxide and the bacterial response to nitric oxide in both Salmonella typhimurium, which, pretty close to E. coli, not too hard about to work on, but also Staphylococcus aureus, which is a considerably more challenging model to work on, and sort of bridging, unlike many bacteriologists, both the gram-negative and the gram-positive worlds. And he's continued on at Yale in the laboratory of Yale MacMicking, and he's going to share with us today what he's been up to, working on some really neat stuff with Interferon-gamma, as well as using some really modern tools such as Cas9 RNA-seq. And during the time he's been in Yale MacMicking's lab, I'd be remiss if I didn't say he received the very prestigious Hanna Gray Fellowship, which is an incredible honor. So I will let him take it away from here. Looking forward to hearing your talk, Rudy.
All right, thank you, Alistair. That was very kind and generous. And thank you all for this opportunity to come share my research and to hopefully receive some feedback on my ongoing project. Today, I'm going to talk about two host antimicrobial defense mechanisms against bacterial pathogens. I'll briefly talk about my project in Ferric Fong's involving nitric oxide. This is a classic host defense molecule, and what we found in terms of its antivirulence role against Staph aureus, as Alistair mentioned. And then the later part of my talk, I'll talk about what I'm doing in John's lab, trying to characterize new actin-based defense mechanisms. More specifically, I'm going to talk about XRP1, which is an actin-binding protein that is induced during inflammatory processes. And I'll tell you about what we think its role is in host pathogen interactions. But before I get into the science, I do just want to take this opportunity to thank the UCSD research community. As Alistair said, this is pretty much where my research career started. Hopefully, this is very familiar to many of you. It's the Scripps Pier down in La Jolla. And actually, this is where I started research collecting marine water samples just off the pier in the laboratory of Brian Pelnick. So I do want to thank Brian for getting me excited about environmental microorganisms, and then later on Victor Nisset, who got me excited about microorganisms that cause human disease. So thanks everybody at UCSD. All right, so a little bit about myself. As a scientist, my primary goal is to understand the impact that microorganisms have on human health. And most of my work has been done on bacterial pathogens. I'm interested in organisms that are beneficial to the human host. Also, bacterial commensal organisms like Streptococcus pyogenes and Staph aureus that live in the human body and for the most part don't cause any harm to our health, but can suddenly turn pathogenic and cause a variety of life-threatening infections. More recently, I've also gained an interest for pathogens that we can acquire from the environment through contaminated food and water sources. So these include bacteria like Salmonella, Listeria, and Shigella. My goal is also to understand the host side, so how the immune system can detect the diversity of bacterial pathogens that is out there, and how it elicits specific responses to help clear these bacterial infections. And so a large part of my work is focused on understanding host defense mechanisms that are mediated by an important immune molecule, Interferon-gamma or type 2 interferons. So the primary reason why I'm interested in studying infectious diseases is because they contribute a large portion to the overall global disease burden, and also they disproportionately affect the most disadvantaged members of our society, low-income communities. And so this is something that has become quite evident today in the time of COVID-19, but we've known for quite a long period of time. So interferon-gamma is incredibly important for controlling microbial pathogens. And so not only bacteria, also parasites and viruses. It is a cytokine that is produced by immune cells such as T cells and NK cells, and it helps the host defend itself against microbes by stimulating thousands of genes to be expressed both in non-immune cells like fibroblasts and also in professional immune cells like macrophages. Interferon-gamma binds the interferon-gamma receptor, and it signals through the JAK-STAT signaling pathway. This induces phosphorylation of STAT1, dimerization, and translocation into the nucleus. In the nucleus, STAT1 interacts with gamma-activated sites upstream of interferon-stimulated genes. And so as you can see in this RNA-seq data from our lab and also from publicly available databases, there are thousands of genes that are differentially regulated. Some are down and some are upregulated. And so the two defense mechanisms that I'll focus on today include nitric oxide synthase. This is the gene that codes for an enzyme that produces nitric oxide. This is a defense mechanism that predominantly functions in macrophages. And then later I'll talk about XRP1, immune cells, and professional immune cells. So among these thousands of proteins that are induced during infection, we can find a variety of classic antimicrobial molecules like reactive oxygen species and reactive nitrogen species. Also, guanylate binding proteins, which my lab currently studies here at Yale. John MacMicking has been studying GBPs for a few decades now. There are also important lysosomal proteins that function within the lysosomal pathway to degrade bacteria and other pathogens. And I'm recently interested in these actin-binding proteins that we know quite a bit about their interactions with actin, but less about how they function in a host defense. So I'll start with reactive nitrogen species such as nitric oxide. Nitric oxide is a molecule that is produced during infection to help fight microbial pathogens. It is stimulated by interferon-gamma and also by pathogen-associated molecular patterns like LPS and lipoteichoic acids. So these are sensed through toll-like receptors. And when macrophages sense inflammatory stimuli or pathogens, they induce the expression of iNOS, which uses L-arginine and molecular oxygen to generate one molecule of nitric oxide. Nitric oxide can be incredibly toxic to a variety of microbial organisms and also to the host itself. But nitric oxide is mostly expressed within compartments of the cell that don't harm the host, but do harm the invading microbial pathogens. Nitric oxide is a reactive radical. It has specific reactivity with metal centers. It can target iron that is found in heme and as well as non-heme iron. It can interact with iron sulfur clusters. These are incredibly important redox centers in a variety of metabolic enzymes. And then lastly, it can also target thiols, which are found in cysteine residues of proteins. And so over the last few decades, we've begun to understand how nitric oxide in general works as an antibacterial molecule. We know that it has pleiotropic effects on pathogens, and it functions mainly through a bacteriostatic mechanism by shutting down metabolic processes like the TCA cycle, the respiratory transport chain, DNA synthesis, and also disrupts iron homeostasis. And most of these interactions are directly by targeting metal centers within important enzymes in these pathways. So in a Ferrex lab, I became interested in studying Staphylococcus aureus, and mainly because in contrast to many of the bacteria that are inhibited by nitric oxide, Staph aureus can actually grow in the presence of high levels of NO. Staphylococcus aureus asymptomatically colonizes approximately a third of the human population, and one of the primary environments is the human nose. In the human nose, we are constitutively producing nitric oxide. This comes from epithelial cells. And so we were very interested in Staph aureus because it has this sort of duality where it lives on the human nose as a commensal, but it can also be a very dangerous opportunistic pathogen causing life-threatening infections at other anatomical sites. We were interested in seeing if there's a connection between nitric oxide, which is expressed in the nose, and staph is not harmful, and a very important microbial communication system called the AGR system. This is responsible for the production of toxins by Staph aureus, and it's quite central to the pathogenesis of this bacteria. So using mass spectrometry and other biochemical tools, in vitro, we were able to show that nitric oxide does in fact interact with the AGR quorum sensing system. It specifically targets cysteine residues within this AGRA transcription regulator, and modification of this cysteine residues shuts down the ability of Staph aureus to use quorum sensing. So normally there is a peptide that is secreted by Staph aureus at high cell density. It is sensed by a sensor kinase, which activates the transcription factor AGRA. And in the presence of nitric oxide, we can see that transcription activation of important toxins such as the phenosoluble modulants. This is inhibited, and also nitric oxide inhibits an important regulatory RNA called RNA3, which post-transcriptionally activates other toxins. And so inhibition by nitric oxide seems to be quite specific to the system, since housekeeping genes are not inhibited by NO. So as I mentioned, the AGR quorum sensing system is critical for staph pathogenesis. With staph is living as a commensal organism, it mostly expresses adhesion molecules in the nose that are important for colonization. There are slow level expression of the quorum sensing signal. However, when there's disruptances in the environment leading to higher cell density of Staph aureus, staph will ramp up the production of the quorum sensing signal, which at a particular threshold, it will activate the production of toxins like alpha toxin, leukocidins, and these phenosoluble modulants. This is data from previous publications showing that nasal carriers that are asymptomatic have an AGR system that is mostly found in an off state, along with low levels of toxin. However, we've known for a long period of time that AGR is incredibly important for pathogenesis. And so in this murine abscess model, we can see that if we infect with wild type bacteria, there's a lot of morbidity associated with these types of infections. But if there's Staph aureus that has a non-functional AGR system, a lot of the pathogenesis can be prevented. So this is data from Richard Novick's group, and so we wanted to test also whether nitric oxide, as we saw in vitro, can inhibit the production of toxins in a whole organism. And so we decided to use this murine pneumonia model because it is fairly well characterized. So a lot of the damage that is associated during staph pneumonia is attributed to one toxin. This is alpha toxin, which is under the regulation of the AGR system. And so here we're comparing C57 black 6 mice in parallel with mice that do not produce nitric oxide. And we were actually able to see that mice that don't produce NO and therefore allow higher activation of the AGR system show higher levels of alpha toxin in the lung. And we were also able to link alpha toxin production to the AGR system. If we mutate the cysteine residues to serine residues in the transcription factor, and then infect with those mutant bacteria, we see elevated levels of alpha toxin. So nitric oxide does seem to have a role in suppressing the production of toxins in the lung. Mice that don't produce nitric oxide also show higher levels of inflammatory molecules like IL-17, which is important for mucosal immunity. Also, CXCL1, which is a neutrophil chemoattractant. We found higher levels in INOS deficient mice. We saw differential histopathology with INOS mice showing a more dispersed pattern of infection compared to wild type. And so ultimately, mice that don't produce NO succumb to staph pneumonia at higher rates compared to wild type. So this mechanism that we uncovered of nitric oxide inhibiting microbial communication is quite distinct from what has been previously found with how NO functions as a host defense molecule. So we've known for quite a long period of time that NO inhibits the growth of microbial pathogens. But now we understand that NO can also work against microbes like Staph aureus that grow in high levels of NO by functioning as an antivirulence molecule. Okay, so so in microbial infections, toxin production is one aspect of infections. In the next part of my talk, I'll talk about the role that, or the importance of recruiting immune cells to the site of infection, and also bacterial spread through tissues during infections. And so this is these are biological processes that are mediated by actin-binding proteins. And so why did we become interested in studying actin-binding proteins? Actin is incredibly important for the physiology of the eukaryotic host. Together with intermediate filaments and microtubules, it forms the cell cytoskeleton. Actin polymers can be assembled into branch filaments or also into elongated fibers, as seen in microvilli, filopodia. These polymers help organize subcellular compartments and also give the cell its shape. Actin is involved in fundamental biological processes like cell division, cell motility, and also allows for the formation of cell-to-cell junctions. And so while actin is incredibly important for the eukaryotic host, microbial pathogens have evolved mechanisms to use actin polymerization for their own advantage during infections. This is an example of an extracellular pathogen, enteropathogenic E. coli, which uses host actin to raise these actin-rich pedestals at the surface of epithelial cells in the gut of mammals. We also know of intracellular pathogens that use actin polymerization to gain access to the host cytosol. These include bacteria like Shigella, Salmonella, and Yersinia that use usually type 3 secretion systems to inject effector proteins that manipulate actin binding proteins, ultimately leading to polymerization of actin and the internalization of the pathogen. Later on, I'll talk more about Listeria, which is another pathogen that is also invades host cells. But in addition to invading cells, it has other mechanisms to spread from cell to cell. Listeria, along with Shigella, Burkholderia, and Rickettsia, can recruit actin polymerization machinery within the host cytoplasm to move within the cell and spread to neighboring cells. So I'm going back to the RNA-seq data just to show you there are dozens of proteins, or actin-binding proteins, that are stimulated by interferon-gamma. So among the thousands of genes that I showed you, we were able to filter out those genes that are associated with actin functions using our gene ontology terms. And so we decided to focus on this gene, XIRP1. because it is very highly induced in both macrophages and also in non-immune cells, such as fibroblasts. And additionally, we know that XRP1 can directly bind actin filaments. XRP1 stands for Chin Repeat Protein 1, and it's a gene that comes in 3 different isoforms. There's a full-length version as well as 2 truncated versions at the N and C terminus. 2 of these isoforms contain what are called the Chin repeats. There are 17 in humans, and these are directly involved in binding filamentous actin. Chin means heart in Chinese, and so this protein was originally named for its constitutive role in the heart, where it's associated with specialized cells-to-cell junctions called intercalated discs. However, XRP was particularly intriguing because it really has no previously known role in host defense, and is not known to be involved during microbial infections. So we were wondering what this heart protein was doing in the context of inflammation and microbial infections. A closer look at the phylogenetics and gene syntheny of XRP does suggest that XRP might be involved in host defense and immune processes. XRP proteins are found in a wide range of organisms, ranging from sea lamprey to mammals. And very early on, we could see that there is a genetic linkage with MIT88, which is an important innate immune adapter protein involved in cell signaling. And then within mammals, in particular within human chromosome 3, we also see that XRP seems to be associated with these chemokine receptor clusters. So most of chemokine receptors are found within chromosome 2 and 3. There are 2 clusters in this chromosome, and XRP is found right next to CX3CR1, which is a chemokine receptor that is used by immune cells like macrophages to move to inflammatory sites during infections. So to try to understand when is it that macrophages express XRP1, we decided to take advantage of the vast amount of RNA-seq data that's publicly available. And so here we are looking at almost 2,000 samples within macrophages containing various conditions. The first thing that I want to point out is that XRP1 is expressed by THP1 cells. These are cells that are used as a model to study monocytes and macrophages. When these cells are differentiated to macrophages and polarized to inflammatory to an inflammatory phenotype using LPS and interferon-gamma, this induces even higher levels of XRP1. Aside from cell lines, we also see that XRP1 is expressed from human monocyte-derived macrophages that have been infected with gram-negative organisms like Salmonella and gram-positive bacteria like Listeria monocytogenes. There are also other bacteria like Mycobacteria tuberculosis, Bovis, and Schmickmatis that also seem to induce macrophages to express this protein. So here we are looking at monolayers of fibroblasts. So as I mentioned, XRP1 is also expressed in these non-immune cells. And while we can see high levels of XRP transcript through RNA-seq, at the protein level, the regulation of XRP is slightly more complicated. So here we have a panel of inflammatory cytokines, and no individual cytokine leads to the production of XRP1, despite that we see these high transcript levels. And so it's the combination of interferon-gamma with IL-1 beta, which is produced by macrophages, that leads the entire monolayer of fibroblasts to express this protein. Here we see no signal with our CRISPR-1 knockout, and through Western blot, we're able to detect at least 2 of the isoforms. And so these are the bigger isoforms containing the actin-binding repeats. We see something very similar with macrophages. However, in macrophages, interferon-gamma does lead to some production of XRP1, where approximately half of the population of macrophages express this protein, but the combinatorial treatment with IL-1 beta engages most of the population. And again, we can see 2 of the isoforms being expressed in macrophages. In the absence of IL-1 beta, we also can detect the expression of XRP1 in combination with bacterial pathogens. So here in green, we're infecting these monolayers with either Listeria, Shigella, and Salmonella. And so these are infection foci that have started with a single bacteria. The bacteria has replicated and spread to other neighboring cells. And you can see that XRP1 or fibroblasts express XRP1 mostly around where the infection foci is. This is true for Listeria, but with gram-negative organisms, despite having the smaller infection foci, seem to engage the entire monolayer. So there's probably some differences in terms of stimulation with different pathogens. This is our current regulatory model of how we can achieve optimal XRP expression. Obviously, the interferon-gamma pathway seems to be critical. And we can either combine interferon-gamma with IL-1 beta produced by macrophages during infection, or by adding bacteria. Both of these receptors signal through MIT88, which activates NF-kappa B. And by looking at the regulatory region of XRP1, we've been able to detect various predicted binding sites for both STAT1 and NF-kappa B. So multiple inflammatory stimuli seem to be required for optimal XRP expression. A closer look at where XRP1 localizes within macrophages actually yielded very exciting results. We found that a lot of XRP1 is found in association with these circular structures that you see here. These are adhesion structures used by macrophages. They're called podosomes. Through confocal microscopy, we can see that these podosomes are mainly localized at the base of the cell, where it makes contact with the substrate. So here's the nucleus, this is the cell, this is the bottom of the cell, and podosomes are also known to have this very peculiar conical shape. So you see all these cones at the bottom of the cell. Podosomes consist of a filamentous actin core, shown here in green. This actin core is associated with actin-binding proteins, and there are also other actin-binding proteins surrounding the podosome. And so we see that XRP1 actually is on the outside area of the podosome surrounding these individual actin-rich structures. This is a 3-dimensional representation of where we see XRP1, again, at the base of the cell in these conical shape podosomes. So now we have also looked at which particular isoforms are required for localization to these actin structures in macrophages. And we've actually found out, to our surprise, that the individual isoforms by themselves can actually be recruited to podosomes. So here we're first using our CRISPR knockout and then using retroviral transduction to add back the individual isoforms. And as you can see, when we probe with an antibody to the N-terminus, that smaller, or actually mid-sized isoform at the N-terminus is sufficient to localize to the podosomes. These are the specks that you see on the cells. But also the C-terminal region, which does not have the domains that interact directly with actin, but does have domains that interact with other actin-binding proteins, seems to be sufficient to localize to podosomes. So again, we're using our knockout and reintroducing the isoforms. And so here at the C-terminus, this is sufficient for localization to podosomes. Now, we would also like to understand the specific location of XRP1 within podosomes to get insights into possible function for XRP1 within macrophages. And so we're using a super resolution microscopy to look at XRP1 in association with other podosome proteins. So again, here are the orthogonal views showing filamentous actin with XRP surrounding this actin-rich core. And here with greater detail, we can see that XRP1 might also be mediating interactions between potosomes, forming this potosomal network. Another potosomal protein that we've looked at is vinculin. Vinculin shows a slightly different pattern of immunostaining compared to filamentous actin. So actin's mostly at the center, and so here when we stain with vinculin, we don't see signal at the center, but we actually see these donut-shaped circles surrounding the filamentous actin core, and XRP is even further outside of vinculin. So where we think actin is within potosomes is right outside that filamentous actin core. We think it's associated with these lateral fibers that are usually composed of non-branch actin, and possibly also these connecting filaments that connect potosomes to one another. Vinculin, the one that has this sort of circular donut shape, is a protein that links actin to membrane proteins like integrins. And so we see it in this adhesion ring that has been previously described. So XRP, we think mainly is involved with the cap, which is thought to function in generating contractile forces that allow cells to move. So we are continuing to use super resolution microscopy to understand the associations of XRP1 with other potosomal proteins. And in the future, we hope to use cryo-electron tomography with instruments that are available here at Yale to gain greater insight into the structure and biophysics of potosomes. One function that we think XRP1 is incredibly important for during microbial infections is the recruitment of macrophages to the site of infections. And so we think that this is true because of the localization of XRP1 to potosomes, and also because it's induced by such a wide range of microbial pathogens. Potosomes are known to be very important for macrophage chemotaxis, also extravasation, or the movement of cells from blood vessels into tissues. They can probe for weaknesses in the endothelial layer, are also involved in the formation of these invasive structures that allow macrophages to move between blood vessels. They are also known for delivering proteases to degrade the extracellular matrix, and of course they're important for attachment to the extracellular matrix. So we are also investigating macrophage transmigration as well as chemotaxis using these commercially available chambers. I think that XRP1 might be particularly important for helping macrophages move within constricted spaces. So these are images that I have captured of potosomes and XRP1. Here you can see one macrophage clearly squeezed between adjacent cells, and you can see potosomes at opposite ends of the cell where they might be mediating adhesion, and also through contractile forces allowing the cell to pull itself through these gaps. In previous studies, it is known that potosomes assemble on filters. Specifically, they form on these filters, and if these filters have gaps big enough, they mediate cell movement through the gaps. Another important process that might be mediated by XRP1 might be filopodia-based phagocytosis. So a lot of us are familiar with lamellipodia-based phagocytosis, where the entire cell will move to an antigen. But macrophages are also capable of filopodia-based phagocytosis, where the cell body will mostly stay stationary, but the cell will actually extend these projections to the antigen. These projections are usually consist of these elongated actin fibers and proteins like VASP, And as it turns out, VASP is actually an interacting partner of XIRP1. So we are quite interested in investigating filoprotein-based mechanisms of antigen uptake. Okay, so in this next section, we wanted to look at the role of XIRP1 in association with bacteria. And so we chose to use Listeria among the wide range of pathogens that induce XIRP1 because it actually uses or hijacks actin to move within the cytoplasmic environment. Listeria can either invade cells or it can be taken up by professional phagocytes. Listeria is very good at evading degradation through the lysosomes because it can express virulence factors that allow it to rupture the phagosome. When Listeria escapes into the cytoplasmic environment, it then uses this protein ACT-A to recruit actin polymerization proteins that will allow it to move within the cell using actin-based motility. And so in macrophage infections, we have actually detected XIRP1 in association with a subpopulation of bacteria. So not all the bacteria within the macrophage are actually coated by XIRP1, but some of them actually form a very nice coat around the bacteria. And these bacteria are completely encapsulated by XIRP1. Coating of bacteria by XIRP1 seems to occur around 3 hours post-infection, so probably not during the initial processes that occur when Listeria is being internalized, at least in this context. And so we think that XIRP1 coating occurs once Listeria is within the cytoplasmic environment. We know that XIRP-positive bacteria are not associated with lysosomes. When we probe with LAMP1, which are lysosomal markers, LAMP-positive bacteria are never associated with XIRP1, and XIRP1 positive bacteria are never associated with LAMP1 markers. So Listeria is definitely not within lysosomal compartments. We've also used genetic tools to knock out important virulence factors in Listeria. If Listeria cannot rupture the phagosome because it's missing these virulence factors required for rupturing, we never see coating by XIRP1. We can also infect with Listeria that do escape into the cytoplasm but do not recruit host actin. So these are ACT-A mutants. These bacteria also are never targeted by XIRP1. So only wild-type Listeria that can escape into the cytoplasm and recruit host actin is able to be targeted by this protein. So now we're beginning to use live cell microscopy to understand or to begin to understand the significance of this coat around the bacteria. This is a macrophage that has 2 intracellular Listeria coated by XIRP1. And here we can monitor for quite long periods of time. And we don't see any growth of the bacteria, but we also don't see any degradation. So you could actually see the photobleaching that occurs over time. But these bacteria are not really growing. They're not being degraded. In some circumstances, we do see that Listeria can actually replicate when it's coated by XIRP1. So these 2 bacteria you'll see replicating and having a light XIRP1 coat. We could see, I'll play it one more time. So here they're replicating. At the end of replication, they seem to undergo this bend, and we've actually captured them escaping the XIRP1 coat. So this is after replication, they do this bend and they'll escape right through the middle, as you'll see here. So there they go, they leave the XIRP1 coat. XIRP1 also localizes to the tails of Listeria as they move throughout the cytoplasm of the macrophage. So you can see the actin-rich tails behind Listeria as it's using actin-based motility. Lastly, we also see that XIRP1 localizes to bacteria at the periphery of cells right before Listeria can actually escape the macrophage. So you'll see this bacteria leaving the macrophage and leaving behind this XRP1 coat. So currently, we don't completely understand whether this XRP1 is beneficial to the bacteria or if it's truly a host defense protein. So we have two different working hypotheses for the function of XRP with Listeria infections. One involves Listeria hijacking XRP1 from potosomes to make actin-based motility more efficient in these inflammatory cells, and might allow Listeria to escape outside of these cells. However, the opposite might be also true, where XRP1 is truly functioning as a host defense protein that might be slowing down actin-based motility and possibly forming these intercellular microbial traps. This would be more consistent with those bacteria that are trapped for long periods of time. This could be an important cell defense pathway if those coated bacteria can then be targeted to other defense pathways such as autophagy. So in the future, we are currently working on measuring things like cell speed within macrophages, both in the presence and absence of XRP1. Also measuring the length of actin tails behind Listeria and the direction of movement. And so by gaining some quantitative data into what's going on with bacterial movement in the presence of XRP1, we'll be able to tell whether this is a protein that is being hijacked or is actually a protein that's functioning as a host defense system against pathogens that use actin-based motility. So to summarize the findings here, we think XRP1 is a novel actin-based defense protein. We think one of its function is to help recruit immune cells like macrophages to the cell, to the site of infection. And so I'll remind you that this is stimulated by a wide range of pathogens, and it has localization, or XRP is localized to potosomes, which is incredibly important for cell movement. With Listeria, we're trying to understand whether this is beneficial for the microbe or for a host during infections. And soon, currently breeding XRP knockout mice to infect with Listeria and other pathogens that don't use actin-based motility to really understand the role of XRP in host immunity. With that, I'll just summarize what I told you today. I talked about nitric oxide, which is a host defense molecule that we found can shut down the production of toxins in Staphylococcus aureus. This is a new mechanism that is independent of microbial growth inhibition. And I talked about XRP1, which is part of my ongoing work, and we think it's involved in recruiting immune cells to the site of infection, and possibly also helping protect against pathogens that use actin-based motility like Listeria and Shigella. So with that, I'd just like to acknowledge my mentors, Ferric Fong at University of Washington, for his guidance with the nitric oxide project, two very talented undergraduate trainees, Helen and Bianca, who helped throughout the project. Also, my current mentor, John MacMicking, for the flexibility and liberty to start this XRP1 project. Also, some very helpful coworkers, Ryan for everyday scientific conversations, Agnieszka, who helped with initial super resolution microscopy tools, Kyle for help with computational biology tools, and Alex, who's now moving on to med school and help build reagents for the XRP1 project. And so, of course, also the funding resources. So with that, I think I'll take any questions, and thank you for your attention.
Thanks, Rudy. Does anyone have any questions at this time? There's a raised hand. Daniela.
Hi, Rudy. Thank you for the very nice presentation. Hi. So I actually have a question. So it was very interesting talk about this new protein that is involved in actin-based motility. And I was wondering, as you know, many of these defenses, even though they've been studied with pathogens, they might actually be more effective against commensals. You know, all these pathogens have a mechanism, for example, to escape. You show like Listeria escapes in the cytosol, Salmonella stays in a vacuole, and they're all protected, and somehow there could be virulence mechanism. But I wonder whether you tried to use some bacteria that actually does not escape, like something like a commensal E. coli, or, I mean, you know, people in the inflammasome field, they've used other bacteria that are actually not able to hijack the host, and this way they actually show a more potent response. So I was wondering what happens if you use a simple E. coli or something that can get killed. Did you still see this accumulation? Do you still see the formation of these, pseudopods?
Yeah, so we haven't tried anything like E. coli. Currently, we sort of started focusing with pathogens that use actin-based motility. So we tried Listeria, we tried Shigella, we tried Burkholderia, and so currently we see only association with Listeria of XIRP1 when it's in the cytoplasmic environment. We are curious about other bacteria that use actin-based motility because there are different mechanisms that these microbes use to actually move. So we want to understand if it's a particular type of proteins that are recruited to actin that XIRP1 can act on. But then also, like you said, I'm curious about Salmonella because it has such a high stimulation for XIRP1, and Salmonella doesn't. Maybe in some cells it does escape into the cytoplasm, but it doesn't use actin-based motility. And there's actually a very recent paper describing the sort of transient actin coat outside of the Salmonella containing vacuole, and some actin binding proteins are important for sort of maintaining the integrity of that Salmonella coat. And when they get rid of those proteins, there is more Salmonella that reaches the cytoplasm. But yeah, of course, I think it'd be good to test a wide range, including more commensal bacteria, and see what roles XIRP1 might have against these bacteria. Definitely.
My second question, sorry, just a quick follow-up. So in your knockout mice, are these full knockouts, or are you doing conditional knockouts in macrophages? What type of experiments are you planning?
Yeah, the mice, it's a full knockout, where it actually has two of the isoforms knocked out. There's a very low expression of the very small C-terminal truncation, and so generally, so these mice don't have phenotypes until they actually get quite old. So as I mentioned, XRP is associated with normal heart function, and so when these mice get old, they do develop some heart issues. But normally, infections are sort of done earlier on, after like around 12 weeks, and so. Yeah, but they are for the most part is a full knockout. Yeah.
Thank you.
Are there any other questions, particularly from trainees? If not, I have a few, as always, but all right, Rudy. So there's all these isoforms. Did you look in your RNA-seq data to see if there's differential responses of these isoforms to your interferon challenge?
Yeah, so I haven't, not in great detail in terms of like how various conditions might affect the production of one isoform over another. All I could tell you at this point is that in the context of just interferon, it seems like the medium isoform, let me see if I could just show you, so you have a visual. Yeah, it seems like the medium isoform is expressed at higher levels. And then it's followed by the full length version and the smaller isoform. And so we are curious as to how like different isoforms might regulate one another. Like you can envision that maybe if there's a protein that both of these isoforms bind to, one might be expressed under some conditions to inhibit the association of the other protein. So yeah, I think we're interested in how they're differentially expressed across different conditions, but also during the same condition. So we're planning on using these fusion proteins to look at multiple isoforms in the same context, whether that is within the potosome or when they're surrounding bacteria like Listeria.
Jack's got a question, so if you want to unmute.
Yeah, I have a question. I think you had a slide where you showed expression outside of macrophages, like in a fibroblast or something like that, induced by interferon IL-1. I was wondering what you think its role is outside of macrophages and non-immune cells.
Yeah, so I think the primary function or how XIRP evolved was probably to mediate. Sorry, I went too far back. To mediate cell-cell interactions and cell movement, and so fibroblasts are actually very motile cells in tissues, and so they're found in a wide range of tissues, and they actually do a lot of they rely on actin fibers, stress fibers, to manipulate the extracellular environment. So they interact with the ECM. They can actually pull using these actin fibers, the extracellular matrix. So you might be familiar with wound healing processes where fibroblasts are very important in actually generating these contractile forces that help close a wound. So I think fibroblasts or XIRP1 might allow fibroblasts to more efficiently remodel tissues during infection. And there's some very interesting data that actually the pattern of the extracellular environment might improve the efficiency of recruitment of immune cells. And this is kind of all dependent on the fibroblast remodeling.
So do you see it induced or constitutively expressed in cancers?
I think it's associated with some cancers. I think head and neck cancers are, but I haven't looked in great depth. But that'd be interesting to see what it might be doing in those types of cancers, and if they're associated with, I don't know, maybe metastatic processes. Yeah.
Great, thanks.
All right, Fabian's got his hand up if you want to unmute Fabian.
Hi, Rudy. Really cool work. Can you go back to the slide where you showed alpha toxin levels and the NOS2 knockout mice? So I was just wondering, so here, did you look at CFUs at all? And do CFUs correlate with those levels of alpha toxin? So is this inhibiting the bacterial growth, and then is this proportional to that, or is it independent of that?
Yeah, so we did a lot of these experiments early on under conditions where we didn't see a lot of differences in CFUs. So we did look at CFUs. But what we were really trying to get at is that, this is a pathogen that's not dramatically affected by NO. And so we didn't, at these conditions where we see differences in the level of toxin, we don't see significant differences in the CFU. So we're attributing nitric oxide function to its ability to inhibit this AGR system and toxin production. So, but we looked at CFUs and the differences are not dramatic compared to the differences in inflammation that we see and the differences in actual toxin present in the lung during staph infections. But yeah, I'm sure that if we looked later on in time points after there's been a lot of lung damage, likely ionized deficient mice would be overwhelmed by growth of bacteria.
I have one last question then to bring us home, Rudy. So you showed the prevalence of this protein in sort of multicellular vertebrates lineage, right? A lot of these actin sort of motility-based pathogens presumably are really, really common in amoebal communities. Do you actually see this if you go outside of the sort of multicellular lineages in the eukaryotes? Do you see this in amoebal lineages, or is this pretty much like a metazoan innovation? Or
Yeah, so XRP in particular is not really found outside of, the sea lamprey, the most sort of distant from mammals where it's found. Yeah. But yeah, there might be a lot of proteins that have very similar analogous functions where they might be important for other unicellular eukaryotes. So that might have similar properties with regard to the interaction with actin. So like bundling properties or the ability to stabilize these actin filaments.
thanks, Rudy. Well, I think unless there's any last minute burning questions, thank you so much for giving a talk, Rudy. I quite enjoyed it. And yes, thank you all for attending. Yeah.
Thank you all for the questions and the feedback.
I'm going to stop the recording. There's the recording button.